Allow me to present our speaker, Dr. Carlos Castro. A little bit about the bio of Dr. Castro. Professor Castro received his bachelor's degree and master's degree in mechanical engineering from both Ohio State University, and then he received his PhD from MIT. He was post-doctoral fellow at the Technische Universität München, working in structural DNA nanotechnology. Dr. Castro joined OSU in 2011 as a faculty member and is currently a professor in the Department of Mechanical and Aerospace Engineering and a faculty member of the OSU Biophysics Graduate Program. His research focuses on self-assembly of DNA nanodevices for biophysical and biomedical applications. Dr. Castro's lab has pioneered the use of engineering mechanics and design approaches for the development of DNA nanomachines with complex motion, dynamic behavior, and program stimulus response. A major goal of his current research is to develop nanodevices for biomedical applications, including diagnostics and drug delivery. He has received honors, including OSU Research and Teaching Awards, an NSF Career Award, Alexander von Humboldt Fellowship, and Fulbright Research Scholar Award. So, Dr. Castro, the floor is yours.
Thank you, Khalil, and it's my pleasure to be here with everyone today. I'll go ahead and share my screen. Are the slides showing up okay?
Yeah.
Okay, great. So yeah, thanks again. Thanks for the nice introduction and invitation. Khalil, it's my pleasure to join you this morning. I'll say sabah al-khair to everyone because it's morning here and also because I don't know how to say good afternoon in Arabic. So I'm exhausted about 10% of my Arabic vocabulary. I'll try the other 90% with the students perhaps afterwards. So it's my pleasure to be here to tell you about, I'll talk a little bit generally first about nanotechnology and how, especially recently in the context of probably the most recent health challenge that everybody's been thinking about, the COVID pandemic, how nanotechnology has played an important role in addressing these societal health challenges. It's a question we're very interested in my lab, developing new nanotechnologies to address biomedical challenges. We also work a lot on fundamental biological, developing tools to answer fundamental biological questions. I won't get into that as much detail, but I'm happy to talk about that perhaps offline or afterwards if anyone is interested. This is our research group here. In the background, you see our department at Ohio State University. It's a beautiful campus, beautiful building. I hope you get a chance to visit in the future. For any students there that might be interested in a graduate program, we do have very strong graduate programs, of course, in engineering, but also in things like biophysics and biochemistry, in case you're interested. And please do feel free to contact me afterwards if you are interested. I'm showing a picture here of our research group. We do have a quite interdisciplinary research group, of course, several mechanical engineers, but we also span areas like biomedical engineering, chemical engineering, biochemistry, biotechnology. And so I think that's really one of the strengths that we bring to these research questions is this a strong integrated interdisciplinary approach where we can draw on several different disciplines to address these questions. So I'll first start with, oh, sorry, I was going to show also, this is a COVID-friendly, of course, we are all dealing with this pandemic and still continuing to deal with this pandemic over the last, several years. And so that's kind of the context you can think about and just point out, this is our group here, again, in this kind of COVID-friendly get-together that we had a couple of years ago. So I'll start with just introducing you to, why is nanotechnology relevant for health or really for biology, right? And we can think about what are the relevant length scales of biological systems if we want to develop technologies to address health challenges. There's, of course, the length scale of a clinical sample, perhaps the length scale of a test tube. which is maybe centimeters to several centimeter length scales. And as we go down in length scale, right, what you see here is a mammalian cell, a eukaryotic cell that's maybe about 10 microns or so in size. If we continue to go down in size, this is a bacteria, right? That's maybe about a micron in diameter, a few microns in length. And if we go down further, right, this is a virus. Viruses tend to be about 100 nanometers in size, right? And of course, the COVID virus is actually, it doesn't exactly look like this, but it looks kind of like this top shell here. That's about 100 nanometers in size. And then if you continue to go down, really a lot of the molecular processes that are driving biological function occur at these length scales where you see proteins. This is a picture of a protein that's about 1 to 10 nanometers in size. Here you have nucleic acid, DNA. DNA has a diameter of about 2 nanometers. I'll be talking a lot about DNA a bit later in the talk. And then ultimately these small molecules, this is a picture of a cholesterol molecule. That's a small molecule that's found in many biological membranes. But the main point here is that a lot of the biological processes, both in normal biological function and in disease, are occurring at these length scales of maybe 1 to hundreds of nanometers, where you have nucleic acids, proteins, viruses, a lot of the important players in driving health and disease. All right, so if we want to make technologies that can influence these biological processes, it's really critical to be able to make devices at these relevant biological length scales that can interact with these biomolecules or with this biological machinery at this 1 to 100 nanometer length scale. All right, and so in terms of technology, right, there's of course many relevant technologies that have been helping address health challenges. One is an example here. I know you can't answer, but I'll give you a second to think about this, what this might be. Maybe you're familiar with this. This is actually the end effector. It's a manipulator that's used on a surgical robot tool. And these end effectors that can grip tissues or move tissues or even cut tissues, right? These can be inserted into a body through maybe a 10 to 20 millimeter incision in the body, right? So this is maybe up at the millimeter to tens of millimeter to centimeter length scale. If we continue to go down, this is maybe not a directly health technology, but is relevant. Again, I'll give you a second to think about what this might be. This is actually a microelectromechanical system. You can see the length scale here is about 20 microns, right? In this particular microelectromechanical system, you actually might be carrying around in your pocket right now. This is an accelerometer, what's called an accelerometer. So this is basically a motion sensor that's used in iPhones and many other device technologies, right? And you see all these blades here. What happens is when you experience any motion or acceleration, these blades deform a little bit, and that changes the spacing between these blades, and that can be picked up with an electrical signal. And so these are, again, very relevant technologies to your everyday life. Another one, this is actually down at the nanometer length scale, right? This is a relatively, maybe 5 years ago now, a picture from IBM showing these nanowires that are built onto transistors. Full disclosure, I know next to nothing about transistors, but just to point out, that the spacings here are about 5 nanometers. So this is really how you pack very densely these wires on a computer chip with these nanotechnologies. The last one I'll show is kind of in between this, single nanometer and micron length scale. And this is really where we're getting to more of the technologies that I'll focus more on throughout the rest of the talk. Again, you can take a second to think about what this might be. I'll say this is something that probably most or all of us have become familiar with over the last few years. This is actually a lipid nanoparticle, and this particular example is an RNA containing lipid nanoparticle, and probably the most well-known examples of these are the COVID-19 viruses or vaccines developed by places like Moderna and Pfizer, BioNTech, their partnership. So this messenger, this is vaccine that many of us have taken, is based on a messenger RNA containing lipid nanoparticle, right? Where the goal of this vaccine, this is about 100 nanometers in diameter, as you saw on the previous slide. Really, the goal of this vaccine is to deliver this, right, to a biological system, to cells, and get those cells to express a molecule that can train an immune response towards the SARS-CoV-2 virus, right? So we want this virus to deliver a material that's going to train your body to ultimately be able to fight future infections, right? And from a nanotechnology perspective, the way that's done is by building these nanoparticles with these particular components, right? There's more here, I'm simplifying, right? But this is the basic idea, right? The main player is this messenger RNA. This messenger RNA encodes a particular protein that's called a spike protein. That protein is found on the coat of a SARS-CoV-2 virus, right? And so now your body is exposed to that protein, it trains the immune system. And now later, when your body's exposed to maybe an active virus, it recognizes that spike protein on the surface, and it can very rapidly mount an immune response, right? Clear the virus before you get sick, basically, right? And the other components here are things like lipids. These contain and stabilize the messenger RNA payload. They might protect it from recognition. They might do things like increase the circulation time or improve the circulation time in the body. There's other components like polyethylene glycol. Polyethylene glycol is a polymer that's biologically inert, arguably biologically inert. This can, again, protect the payload. That's these little squiggly molecules on the surface that can stabilize the particle, protect the payload, again, potentially help circulation, and also maybe protect from things like these foreign vaccine particles introducing an immune response themselves, right? Because what you want is an immune response to the thing that's encoded by the messenger RNA, not to the particle itself. So this is, of course, a very relevant biological technology that's in use today, but this is the product of decades of research. I'm showing this timeline not so you can read all the individual components, but just to stress the point, right, that this has been developed over decades of research, starting as far back as the 1960s with the discovery of messenger RNA and its function. And excuse me, just a few years later, the development of these lipid nanoparticles, and many, many decades of lots of hard work and research, ultimately leading to just a couple of years ago the approval of these COVID-19 vaccines that are these messenger RNA lipid nanoparticles. Right. So so far we said these nanotechnologies can be important tools to help deliver biological payloads. Right. For example, messenger RNA in vaccines, or other people are developing messenger RNA-based therapies. Another way that nanotechnology can be relevant to addressing challenges of health is, of course, through diagnostics, right? This is another critical component of sort of a societal response to a pandemic or to an emerging pathogen. These are, you probably familiar with this test or things like this test. Right, where you take a sample, perhaps you swab a sample from your nose, you put that in one end, right? There's a membrane that sucks up the liquid across this strip, and eventually you see, right? Thankfully, these are actually tests that we took at home, right? You see the control sample that tells you, yes, you have the right kind of sample, the right biological material there, right? And then if it's negative, you don't see a line here. If it's positive, you see a line here. But there's nanotechnology underlying the function of these rapid antigen tests that we've all become probably familiar with over the last couple of years. So how these tests actually work, these are called lateral flow antigen tests, right? And the basic idea here is you put your sample, possibly saliva, possibly blood, right? Possibly a nasal swab in on one end, right? That liquid gets sucked up, right, as it passes across here. When it passes across this conjugated pad, it picks up these nanoparticles. So these pink guys are nanoparticles, right? And if there's the target molecule, this red molecule that will bind to the antibodies that will bind to these nanoparticles. And then if you have this target antigen, right, that'll eventually bind again to these antibodies down here at this particular strip, right? And a different target antigen, right, will cause binding onto this strip here, right? So you have two different targets, one that's just a biological control, something that's inherent in these biological samples to say, yes, that was a good sample. And you have then the target antigen that shows up on this strip here. So if you have a positive test, you have a concentration of these nanoparticles that show up at this particular strip on this lateral flow test. All right, and so again, the goal here is to identify the presence of a disease biomarker. In the case of these COVID tests, that's usually the spike protein or perhaps another protein that's on the shell of the COVID-19 virus. And the way that's done from a nanotechnology perspective, right, you have these nano-sized components, in this case, a nanoparticle that provide the visual signal that are concentrated, right? So here the nanotechnology acts as a reporter, right, to show up as the visible signal on this strip. And of course, the other component here is the molecule that binds the target antigen that we have to conjugate or connect onto the surface of the nanoparticle, right? And that molecule is called an antibody, right? The job of an antibody is to bind the target antigen, right? And basically capture those nanoparticles at this particular location so they become concentrated, and you can see that readout signal. All right, and so this may seem relatively simple here, but probably the challenge is on the system integration, right? Building everything, putting all the components together. And from a nanotechnology perspective, the challenge is really in making these particles and attaching the antibodies here, both to the nanoparticles and maybe attaching the antibodies to the membrane here as well. Right. In fact, this challenge was awarded, technologies to address this challenge was awarded the Nobel Prize in Chemistry earlier this year. This is a group of researchers that developed some methods for biological conjugation. So basically methods to connect biological molecules together, maybe biological to synthetic molecules or materials. Right? And so we can ask the question, why is this difficult really? And that kind of leads to the question of how are these biological nanotechnologies manufactured? Right? And I'm going to go back now to one of the technologies I showed you earlier, this lipid nanoparticle, this messenger RNA carrying lipid nanoparticle. And just show you quickly an animation of how this is manufactured. So if we look at how this is manufactured, this is really done through what's a process called molecular self-assembly, which is a process where we basically put all the components into solution, right? All these components inherently are interacting with each other in some way. And then ultimately these components find each other in solution. So for example, here you have the nucleic acid. That nucleic acid is negatively charged, so it will attract these positively charged molecules. Right now, these positively charged molecules attract other molecules and so on. And so you have these successive layers of molecular self-assembly where you just have components floating around in solution, right? And ultimately you end up with this lipid nanoparticle that contains the payload that you want to deliver, this messenger RNA payload. And one of the questions we're really interested in my lab is, can we use that molecular self-assembly process to build robots, right? To build devices, to build things, right, that we can use for useful applications in biology and medicine, right? These particles, of course, are very useful themselves, but the one thing that you notice is there's not any moving parts here, right? These are static assemblies, right? And then ultimately what happens is they get internalized into a cell, they get degraded, and that messenger RNA gets released into the cell, right? But the question my lab is interested is really, can we build things that operate like this, but can we do that on a length scale, where these robots and devices can interact with those biological molecules, right? And so in other words, right, can we build these complex machines and robots at molecular length scales at this 1 to 100 nanometer length scale I mentioned earlier? And of course, right, we can look to biology for inspiration. Biology builds these exquisite nanomachines. This is an example animation of what's called a molecular motor protein. This molecular motor protein is called kinesin. Its job is to transport materials around inside of a cell, right? And you see this motor protein is about 40 nanometers tall. It walks along these filamentous tracks inside of cells. And basically it transports materials around inside of your cell, right? And if we think about what are the underlying design concepts that maybe we can adapt to developing synthetic nanomachines, right? Here are some of them. The relevant design concepts or constraints, right? These operate at nanometer length scales. There's constant motion here. There's a lot of fluctuation. The water molecules surrounding those machines are moving around, and that drives fluctuation of the molecules, right? These operate at very, very small force scales. We can think about if we want to design a robot, perhaps that can respond to the environment, right? How do these respond to their environment? What are the power sources that drive these nanomachines, right? And so these are many things that we try to draw on for developing these DNA-based nanodevices that I'll talk about in a minute. Where my lab has been somewhat unique in this field is another place we draw inspiration from is actually in macroscopic robots and machines. right? So these are things we can see. These are things that operate in industry, right? These robots, in this case, is an example of a robot that's assisting a manufacturing process, right? It has this robotic arm that can move around with well-programmed motion and a gripper that can grab on to the basket, in this case, move it around, right? It probably has onboard sensing. And so we can think about what the design concepts that underlie these macroscopic engineered machines. And one of the things we do in my lab is really try to combine this inspiration from both biological molecular machines and macroscopic engineered machines and use those design concepts to realize new and useful DNA-based nanodevices. And the first question we can ask here, right, does that make any sense to think about building robots that operate at a molecular length scale that look like robots, right? And kind of giving away the punchline here, I'll tell you, we can build these robots that look like and operate similar to these macroscopic engineered machines. This is actually a little DNA robot arm that's about 200 nanometers tall, right? So not much bigger than a COVID virus, right? And we're able to build something that looks and moves much like this kind of machine here. Right? And throughout the rest of the talk, I'll tell you a little bit more about how we actually do that. And so kind of a general theme of our lab is combining this area of DNA nanotechnology and engineering design for these different application spaces. We work a lot on nanorobotics, as I alluded to, right? We develop tools for biology, meaning thinking about, can we answer fundamental biological questions, right? Just understanding how cells and molecules operate normally. Right? And then, of course, we're interested in developing biomedical nanodevices where we can do things like maybe deliver drugs, right? Or detect biomarkers, right? Markers of disease. And so those are the things I'll talk about today are more in this category, right? So I'll tell you a little bit about how we're developing some nanoparticles for drug delivery and some nanodevices for rapid diagnostics. So I'm going to start with really just introducing you to the field of DNA nanotechnology and kind of introducing you to the basic approach that we use, this DNA origami approach. And then I'll quickly show you just highlights of a couple of projects ongoing in my lab where we're developing nanostructures for therapeutic payload delivery and DNA nanodevices for rapid diagnostics. All right, so the first question you might ask, I showed you earlier, this biological self-assembly, right, where we're using things like lipids, right, charged lipids and messenger RNA, right, to deliver biological material. So a question you might, I'm telling you, we build devices out of DNA, right? So the first question you might think of is, well, why DNA, right? And so the reason is, it turns out DNA is a very useful and convenient material to build nanostructures and materials and devices out of, for the reasons that it follows relatively simple rules for assembly, right? It has a well-known and well-defined structure. It's about 2 nanometers in diameter, right? And it turns out it has tunable mechanical properties, which is something we leverage a lot. I'll come back to that. But first, let me focus on the idea that it has pretty simple rules for self-assembly, right? It may have been a while, maybe or maybe not been a while since you've thought about DNA, but DNA is a double helix, right? It's composed of 2 strands, and the way these 2 strands bind together is that there's 4 different building blocks, right? There's a G base or nucleotide, a C base, an A base, and a T base, right? And the way these 2 strands stick together is that the G bases bind to the C bases and the A bases bind to the T bases, right? And that makes this a relatively simple system to think about designing sequences where we can control how these sequences are going to ultimately bind together, right? And what I mean by that is, with just those simple rules that I just told you, right, we can take, for example, a template sequence strand or a template strand where we know the sequence, right, and just knowing that A binds to T and G binds to C, we could pretty easily write down another sequence, right, that should bind to this template strand, right? Anywhere there's a T, we put an A, anywhere there's a C, we put a G, and so on. If we mix these 2 strands together, we put them in the right conditions, right, we should get this nice DNA double helix, right, which already I think is quite interesting and exciting, that we can actually build these biologically relevant molecules, right, by just writing sequences down on paper. Of course, right, these types of strands typically order from a company that chemically synthesizes them. I'll talk in a minute or two about where we actually get the DNA that we use to build our devices. But the idea to take this basic approach of sequence design and use that to build synthetic nanostructures and materials. That was really pioneered several decades ago by a researcher whose name is Ned Seaman. He's a chemist at New York University. His original idea was to be able to design these sequences such that they would form these junctions, right? So we can design these sequences to pair, right? 4 different sequences to pair together. So you get this 4-arm junction, right? And now what we can do is extend one of these strands by a few bases. So you have what's called a sticky end or an overhang, where you have a few single-stranded bases that can be used to attach to the neighboring junction, as what's shown down here, right? And then we can use that 4-arm junction as a repeating unit to build up these lattices and materials. Right, so really this is where the field started, about 40 years ago now. I'll talk a little bit about how the field has progressed. Right, before that, I'm just going to show you again one of these timelines. I don't put this here so you can read it really. I'm putting this particular one here to highlight another researcher in the field, Hannati Sleeman, who actually has also made major contributions to the field, and actually is a Lebanese professor at McGill University. I believe she got her bachelor's degree at American University of Beirut. So as with many things around the world, I think, Lebanese people out there making strong contributions. And so I just wanted to highlight her. And so coming back to maybe showing, I think, a little bit more visual depiction of progression of the field, right? Here you see where the field started. I won't go through this in detail, but just to give you a sense of how the complexity of these structures have progressed. On the left side, you see just really the geometric complexity, right? Getting to bigger and more complex shapes and structures. On the right side, you see now more the machine and sort of dynamic device side, right? How that has progressed, starting with maybe boxes that could be open and closed to complex machines that can be controlled in real time. And just to highlight a couple of, this is really where my lab has made impacts. A couple of these, this one here, this example here, and this example down here are research that has been developed out of my lab. To give you a better sense of the actual approach that we use to assemble these devices, I'll introduce you to this approach that's called scaffolded DNA origami. This is the particular type of DNA self-assembly that we use in our lab. And in this approach, just like we did before, we're designing sequences, right? We take this template strand. In this case, this template strand is very long. It's maybe 7,000 or 8,000 bases or nucleotides long. This template strand is derived from a bacterial genome or a viral genome. Actually, it's an M13 bacteriophage viral genome. That virus is harmless to humans. It just turns out to be a convenient source that's relatively easy to grow up in the lab. And importantly, we know the full sequence of that genome. Right? And since we know the full sequence, we can design these other sequences that should bind to certain regions of that template strand. Right? So we can design this blue sequence that should bind to the blue here, the green sequence that should bind to the green here, right? And the red sequence that should bind to this red sequence. And the trick here with this DNA origami approach is that now this red, right, blue and green sequences that should bind to these 3 different locations on the scaffold strand or the template strand, we stitch those together onto one continuous strand. And so once I mix these guys together, the only way for those to fully bind is that scaffold strand has to fold up into a more compact shape so that this one continuous strand can bind to these 3 different regions on the scaffold. Right? And expanding that approach out, if we take that scaffold, mix it with many of these strands that we call staple strands because they quite literally pinch together different parts of that scaffold, right? We get this molecular self-assembly process where staples start binding to the scaffold. They're kind of pinching everything together, right? And ultimately, when all the staples bind to the scaffold, you get this nice, well-defined structure that's here kind of shown in a blueprint routing diagram of the DNA. Up here, you see a more realistic depiction of what these structures might actually look like, both as a molecular model and actually a molecular simulation, showing these things are not perfectly rigid, right? There's a lot of waviness, there's a lot of fluctuation, but, right, we can hold the DNA in some target shape, and target desired shape. And this was first demonstrated in 2006 by Paul Rodemund, who's a researcher at California Institute of Technology, and he first demonstrated we can make these interesting structures, things like 100 nanometer smiley faces, not for any particular function, but just to demonstrate, what's the complexity of nanoscale geometries that can be achieved. Right. This is a bit more up-to-date view of the design space of DNA origami nanotechnology, right? With this technology, you can build all kinds of shapes and structures. I would venture to say that just about any shape you could think of, you could probably build something that's relatively close to it, especially with the current recent advances in design methodologies and tools. But here you see we can build these wireframe cages, even things as complex as gears, right? These polyhedra or grid type structures, any kind of two-dimensional shape, right? And really scaling that up to more and more complex shapes and larger structures, right? So there's a huge geometric design space. In my lab, we also think a lot about the mechanical design space, right? I mentioned earlier that DNA has tunable mechanical properties, right? And really the way we do that is we can think about bundling together several pieces of DNA, right? This is much like, bundling together several fibers, right? Or perhaps bundling together several steel wires, right? When you do that, you basically end up with a stiffer system. So what I'm showing here are just thermal fluctuations. These fluctuations are driven purely by the motion of the water. As I mentioned, these systems are small enough to respond to that. That drives the fluctuations, and how much this fluctuates depends on how stiff it is, right? And so just for a piece of double-stranded DNA, you see this is quite flexible. It bends, it twists, it opens and closes, right? It coils up. As we start bundling that DNA into larger cross sections, right, we get things that are stiffer. So here we have 6 pieces of DNA bundled together into this cross section, and you see it's still relatively stiff, relatively flexible, right? But you don't have this thing completely coiling up, right? And ultimately, we can do things like bundle as many as 50 or 56 helices together to get structures that are pretty rigid, right? That are really not subject to these thermal fluctuations. And we work a lot also with single-stranded DNA that's even about 20 times more flexible than double-stranded DNA. And so we have this huge range of geometry and mechanical properties to work with to ultimately build these devices. And so a general theme of our lab, again, is to integrate these flexible and stiff components to design these dynamic nanomachines. Right? And I won't go into much detail about this, but I'll just show highlights really of kind of one of the areas we've really made strong impacts in this field is in nanorobotics, advancing the capability of these devices for nanorobotics and nanomechanical applications, right? And again, I mentioned where we're kind of unique in this field is we take an engineering approach to this design problem. And so we think about designing DNA nanomachines much like we would think about designing a robotic platform like this one, where we need to have precise geometries, right? Like this open frame on the top, but we also need to have these joints that can move in certain ways. So we need to have joints that can rotate, joints that can translate. And we'd like to be able to piece those together to make these more complex moving robotic systems. All right, and so I'll just show you, right, that we can do this. The way we make these rotating joints is we have these stiff bundles of double-stranded DNA, and at one end, we connect them with flexible single-stranded DNA. And what you see here, when we image these by transmission electron microscopy, these are TM images shown here, we can see these guys show up in several different conformations, which indicate to us, right, that these are fluctuating in solution, and when they land on a surface, they get trapped in whatever conformation they were in. On the right here, what I'll show you is these videos. This is not real motion. This is just a bunch of static snapshots that are taken from these types of images that are strung together, but this is to give you a sense of the range of motion, right? So we can build these nanoscale rotary elements, right, really at this 50 nanometer length scale, which again is the length scale of biological molecules and machinery and interactions. And also we can build these translating elements, right? Again, I won't go into a lot of detail here, but just show you some highlights of the other types of devices that we can make. We can make these things that undergo 3-dimensional motion. We have a lot of now molecular simulation to understand how to design certain motion here, certain mechanical properties. And this is one of the more complex, in case there's any paper origami enthusiasts in the audience, right? We've also developed these devices that can change conformation inspired by paper origami designs. This is what's called a water bomb base. And so this is a equivalent DNA nano device that can change shape, right? That can be reconfigured into many different shapes here. And so actually, just in the interest of time, I'm gonna skip over, actually, I'll show you this. Some of the things we're currently working on are really just continuing to advance the design capabilities, right? And sometimes, right, we don't necessarily ask questions that are super relevant to society, right? But in my mind, that's okay sometimes, right? And so we ask questions like, how complex can we get? What can we build? And recently we published a manuscript where we demonstrated that we can get as complex as building, about a 200 nanometer size airplane. And this was an exciting advance, right, from our lab that really is advancing the capabilities of these design tools. We were also excited, actually, this got highlighted on BBC News, so I'll just show you this video quick. And hopefully you could hear the audio there, but that was, again, we were just excited to get this new technology and software highlighted on BBC News about a year ago. I'm going to skip this in the interest of time, and let me jump now. Just in about the next 10 minutes or so, I'll show you a couple examples of how we're taking this DNA nanotechnology and using that again towards health applications, towards health challenges, right? And the first example to try to deliver therapeutic payloads, in this case to leukemia cells. And this is research that started maybe about 8 or 9 years ago. Right? And we were inspired in particular by these two studies that had demonstrated in a breast cancer model system, right, that these DNA nanostructures can be loaded actually with chemotherapeutic drugs. These are anthracycline small molecule drugs that can be loaded onto the DNA nanostructures, right? These can be delivered to cells and they can effectively kill the cells, right? And they actually have some advantages to overcome drug resistance mechanism of the cells. I'll talk about that more in a minute. And this same group demonstrated a couple years later, actually, that this is also effective in a real animal model system, delivering these chemotherapeutic drugs bound to these DNA nanostructures, excuse me, tested in a mouse model system. Right? And so the question we asked is, can we develop similar technologies? All right, but can we do this for leukemia? There's a very active leukemia research group here at Ohio State University in our Comprehensive Cancer Center. And so we partnered with them to try to use this approach, right, to basically deliver drugs to leukemia cells. Right, and so the drug we're delivering is what's called daunorubicin. This is a pretty regularly used frontline therapy for leukemia, acute myeloid leukemia in this case. Right. One of the challenges is these cells often adapt or develop drug resistance. They develop resistance to these drugs where they start expressing these pumps on the surface, where these pumps bind the drug internally inside the cell and can just pump that drug right out of the cell. And so that drug doesn't have time to get to the nucleus and perform its function and ultimately kill the cell. But the idea was here, if we bind this drug to the nanoparticle, deliver that drug-loaded nanoparticle to the cell, that actually gets internalized by a different mechanism into the cell. It gets endocytosed. So as it gets uptaken into the cell, it's trapped in this little, what's called a vesicle or endosome. So that drug is sequestered on the nanoparticle in the endosome, right? And ultimately, it can get far enough into the cell, the drug is released, and we're able to get it to the nucleus, right? And so that was the hypothesis. Right here, what I'm showing you is our drug-carrying nanoparticle. This is a nanorod that's about 90 nanometers long. Here you see TM images and a different type of imaging modality called atomic force microscopy. On the right here, you see particles that are loaded with drug. That's why these particles get a little bit twisted or frayed. It's because you see now the drug is bound. All right, and now we first tested, whether, as we thought, these particles would get taken up via this specific mechanism of endocytosis. And what you see here are these endosomes that are labeled in green. The nanostructures are labeled in red. So where you see yellow, it's basically overlapping green and red signal. And so that does tell us that indeed these nanostructures are getting taken up by these specific mechanism. And kind of the real test here is shown on the right, where we're actually testing the drug delivery. So the fluorescent blue signal that you see is from the drug. On the left column, what you see is if that drug is just introduced free in solution to these drug resistant cells, you don't get much of it into the cell and it gets pumped out over the course of a few hours. Whereas if we first bind that drug to a DNA nanoparticle, now we can much more effectively get that drug into the cell, see a much stronger blue signal in the cell, and that drug stays in the cell longer. And ultimately, this bright signal here is coming from the nucleus, right? And so that's really where the drug needs to go to perform its function. And we were also able to show here that basically delivering these drugs via these nanoparticles, it really does help kill kill these drug-resistant cells more effectively. All right, and so this was a paper we published several years ago now. One of the things we're working on currently is, right, that was a free leukemia cells in culture system. Right, once we introduce this into a real biological system, there's going to be many other cells that these nanoparticles could interact with. Right, and so what we're working on is, can we specifically target these nanoparticles to disease cells, again, in this case, leukemia cells. And the way we're doing that is adding these antibody molecules to the surface here. So you can see here again in these TM images, we can add 1 or 2 antibody molecules. And these are very recent tests just from a few weeks ago. We're able to show, I won't go through the details here, but maybe focus down here. And what you see here is basically the red signal is showing up in the cells that are not labeled green. So here we have our target disease cells that are not labeled green and the off-target cells in green. And so here we're able to show that we can really specifically deliver these nanoparticles to a target cell population in a mixture of cells. We're now performing the experiments to really test, can we perform targeted killing and targeted drug delivery in this mixture of cells? Right. And so just in a few minutes, let me show you again how we're also developing these technologies for rapid diagnostics. Right. In this case, we've been focused on COVID-19 diagnostics, viral diagnostics, right, developing technologies to try to compete with PCR. Right? PCR, of course, is the gold standard. Right? It's great. It's highly accurate, highly sensitive. But the challenge here is it takes a long time. It takes several hours, and oftentimes it might take a couple of days to get a response. Right? And that's really because this technology that's required, this quantitative PCR that's required, this is typically carried out in centralized labs. So typically your sample gets shipped, right, to some other lab. The test is carried out there, and then they send your results to the clinic, right? And then you ultimately get your results maybe a day or two later. Of course, there's other tests, like I showed earlier, these rapid antigen tests. These are great. These are simple, fast, cheap, right? But the accuracy is not great. You may have heard of stories of false positives and things like that. We had a significant case close to home, right, where our state governor actually had a false positive test right in the midst of thinking about buying a bunch of these, right? Ultimately, I think the state of Ohio had a pretty good diagnostic response, right? But this, of course, causes challenges if you have false positives in these rapid tests. And so what we would like to do is sort of develop a test diagnostic assay that has the advantages of both, right? The accuracy of the PCR and kind of the convenience and cheap, easy advantages of the rapid antigen tests, right? And so we're doing that through this idea of making these DNA-based biosensors, where we have these hinge-like devices that are latched. I'll show you in the next slide a quick animation of the function here. We have these hinge devices that are latched. We can add a molecule to disrupt the viral shell. Once the nucleic acid inside is exposed, that can come and release the latch on these hinges, right? And that releasing of the latch allows these hinges to open, and we get a strong fluorescent signal that reports on the opening of the hinge, or basically the presence of that viral nucleic acid. Right, and this is just one piece of data that I'll show you, where you see these nano devices that are primed for sensing, right? They're latched closed. We introduce those right into solution. We have the fluorescent signal from these guys. When we add the target RNA, the viral RNA, you see we get a robust increase in signal within a few minutes, right? Ultimately ending up with these open devices, right? So we're very excited, right, about the prospect of having these rapid diagnostics with these DNA devices. We've been lucky to partner with a local company. We've even tested this on patient samples. And so far we have about 70 to 80% accuracy in detecting positive patient samples. And we're currently working on methods to increase the sensitivity. Our challenge here is really to increase the sensitivity. All right, and so ultimately what we envision here is a single-step assay where we have our devices in solution. You add your saliva sample, right? If the virus is present there, right, then hopefully within about 10 minutes, you can get that response. And this can be read out on sort of cheap and easily available instrumentation. Right, and so I'll skip this. This is kind of where we're going with this, but I'll skip here. And so let me I was going to show this as kind of an overview of some of the directions we're going in, but again, I'll skip that in the interest of time. I'll finish up there and just thank the funding sources. Of course, much of this work has been funded by the National Science Foundation, some of it also by other agencies like NIH or Department of Energy and American Heart Association. Right? And lastly, say shukran to all of you, shukran to Khalil, and of course also maybe special thanks to Khalil and the family. You know, it's not just because it's a wonderful family, but also they've given me the opportunity actually to go visit and experience Lebanese culture, right, and visit Beirut, actually, as you see here. So hopefully next time, I hope I can visit you in person. So yeah, thank you. I'm happy to take any questions.
Thank you, Dr. Castro. Thank you very much. As much as I could understand, it was extremely, extremely interesting, to be honest with you, but thank you. Why don't I open it up? Are there any questions from the floor? Yes, Dr. Faris. Can you open it?
Carlos, I have a quick question about the this nanotechnology. DNA is wonderful, as you showed. I didn't realize you can build such complex structures. But DNA is also unstable in the sense that there are enzymes that degrade it. So I was wondering, when you go to the in vivo model, not in cell culture, do you envision large amounts? And if you do, since you're using viral DNA, it's immunogenic. Actually, DNA, what people don't realize, it can be immunogenic, especially if you use bacterial and viral sources. So do you have ideas about how to circumvent the immune system?
Yeah, no, thanks. Yeah, thanks. Those are both very good and relevant questions. So in terms of the stability question, these DNA nanoparticles are significantly more stable than naked DNA because basically you're packaging the DNA into some compact shape, right? And so these are much more stable against enzymatic degradation. So we and others have done tests where if you put these in serum, for example, where naked DNA might degrade within minutes, these things can last several hours, up to 6 hours, 10 hours. And if you need additional, enhanced stability beyond that, right, you can do things like, again, we and others have developed these methods. I didn't show them here today, but you can coat the surface with things like polyethylene glycol, right, that can improve the stability. And to that point as well, it's also a very relevant point that DNA can be immunogenic. We actually just published a paper earlier this summer, testing the safety profile of these nanostructures in a mouse model. And what we found is we do see a little bit of immune response. It's actually different. So our positive control in that set of experiments was a bacterially derived DNA sequence, and that bacterially derived DNA sequences, as you suggested, elicits a strong immune response. But these nanostructures only elicit a pretty mild immune response, and that immune response subsides within a few days. We tested this over repeated injections, actually 5 high-dose injections over a 10-day time course. But again, we think they're suitable for these relevant, biological and clinical applications because of they are relatively safe, as we found. But if that modest immune response is a problem, again, there's approaches like coating them with polyethylene glycol or even modifying the nucleic acids, chemically modifying the nucleic acids. This is what similar methods to what people do with RNA to again prevent the immune recognition of those molecules in a real system. Yeah.
Thank you. Any other questions from the floor? Okay, this is again.
I actually have a second question for you. This is actually, for our students, might be a little bit advanced, but they shouldn't be ask anything. No question is stupid. That's how you learn. So here's another question. Have you considered using histones or something like that to alter the packaging, or that's too hard? I mean, you're trying to do self-assembly, so it might not be something that's, interesting for you. But to create, even different structures by adding histones to the mix or other DNA binding proteins from bacteria, not essentially histones.
Yeah, no, that's a great question. So there's a whole host of DNA binding proteins that can deform and change the shape of the DNA. And you mentioned histones. let me just go back here. We are actually developing devices to study nucleosomes and these histones. So what you see here, these are actually these histone proteins wrapped with DNA. This is how DNA gets packaged up inside of cells. So we're developing tools to study histones and nucleosomes and their structure and mechanical properties. Because of that, we have kind of been thinking about potentially using things like histones and other things like transcription factors that maybe bind and deform and provide new ways to actuate or control the structure of these devices. So we're currently active in developing tools to study the biology of those molecules, right? But in terms of using those molecules to control or modify the devices or make the devices, it's something we thought about, but we haven't really done just yet. But that's, I think, an exciting idea.
I have a question, Dr. Castro. Tell me, all of what is happening here, how much of it is coming into our part of the world? Do you know, is there a research going on? Is there any interest in all of this, what you have talked about?
Yeah, no, that's to my knowledge. So first of all, I'm sure there is. The things I'm aware of, I have met at conferences a couple of researchers from AUB chemistry program that are interested in nanoparticles. I believe there's a researcher in the chemistry department that's interested in these DNA-based, or not, sorry, these metal-based nanoparticles for diagnostic applications. I know there's other researchers there, I think in some of the hospitals that are working on also drug delivery challenges, not to my knowledge with DNA nanotechnology, but yeah, but I think, you know, other technologies for drug delivery applications. I will say, if there's interest, you know, I'm more than happy to think about collaboration. I didn't talk about it today. We actually we did develop experiments that can be carried out in a classroom. So one of the things we're very interested in is translating this technology into the classroom so that students can learn about it, you know, not just by hearing someone talk about it, but actually by building and characterizing these structures, right? So we've developed methods. I'm happy to share. We do have a publication on that. I'm happy to share that in case anyone's interested on how can you actually carry out experiments in a classroom with equipment that costs a couple $100, where students can actually make and do a little bit of analysis on these DNA origami nanostructures.
Thank you. Any more questions? Dr. Faris, again, it's your turn.
The students are being shy, so and it's a fairly advanced topic, even though you made it sound really simple, but this is a fairly advanced topic for them at this point. In a couple of years, it'll be much easier for them. It's a two-part question. One, besides these Pfizer vaccines, have been applications? And two, are they just for drug delivery? Is anybody thinking about designing them in a way to alter structures, you know, cell structures, or to stitch wounds, for example, or, you know, things beyond just drug delivery?
Yes, yeah. So those are great questions. So beyond drug delivery, so you mentioned vaccines, right? So actually, again, I didn't talk about it because of time, but actually our lab and a couple other labs are interested in developing these nanoparticles as vaccines. Some of the main advantages with respect to vaccines, again, are things like the ability to target to the relevant cells that you might want these, you know, specific viral proteins to go to. You can also deliver a pretty high payload of these viral protein molecules, and it's a very well-controlled system, right? So you maybe have the potential to do things like control how it circulates, right? How long it stays in the body, things like that, just because these are very well-controlled design nanoparticles. And to your other question, in terms of, are there other applications in biology, even things like wound healing? So we are certainly interested in things like controlling cell-cell interactions. Actually, this image right here is a picture of these DNA nanostructures. The fluorescent signal that you see is coming from DNA nanostructures embedded on the surface of a cell. So we're interested in things like controlling cell-cell interactions that maybe are relevant to things like wound healing, or even things like stabilizing leaky vasculature. That's another question we're interested in. Tumor sites, the vasculature often becomes leaky, which is one of the relevant models of disease progression and relevant mechanisms for disease progression and metastasis. So we're developing devices that we can embed at those junctions to help hopefully stabilize those junctions and prevent or reverse that leaky vasculature. Yeah, kind of relevant to wound healing, we are, what you see here are these DNA nanodevices embedded on a cell surface in a 3D tissue. So we are moving towards these more tissue-like applications right now, where we really can try to control what a cell is doing or monitor what a cell is doing. But we do hope ultimately to be able to embed these nano devices sort of throughout a tissue, where maybe we could do things like control signaling, maybe even control collagen structure or tissue structure. And that could certainly be relevant for some of these other applications like wound healing.
Dr. Castro, there is a question in French. I have to ask our dean to interpret this to you, because I can't. Hannah, can you take over this question, at least and ask Carlos?
Sure. Are the stuff that you are presenting, the things you are presenting, will they interest students who will be studying medicine or mechanical engineering?
Sure. So that's a good question. I guess my short answer is hopefully both. I think again, these problems of developing technologies for biological applications and medical applications can certainly benefit from having students both from engineering backgrounds and from medical backgrounds involved in these research projects. We try to do that the best we can. We certainly have engineers involved. We collaborate actively with medical researchers. And so, as an engineer, right, the things I'm an expert in is, how do we build these devices? How do we measure the properties? How do we tune the properties? How do we model them? Right, but I really need advice and input from medical researchers to understand, where can these be useful? What are the mechanisms of disease that we could potentially address? What are the target molecules that might be of interest for a particular cancer? Right? How do we need to design the particles so that they can interact with the cell in a certain way? So I think, the most effective research comes when you can really involve students from these different disciplines.
So as a follow-up, if I may ask, do you anticipate that this will be something that future doctors will be using a lot? I mean, is this something that is going to be a lot of delivery, a lot of therapeutics will be based on these kind of nanotechnologies?
I'll say I'm optimistic. There's nothing, clinically approved or close to clinically approved right now. But what I would say is quite encouraging is there is increasing interest from industry, right? So until about 3 or 4 years ago, this was exclusively research that was carried out in academic labs. But over the last several years, we've had a contact with pharmaceutical companies, with small biotechnology companies that are really getting interested in these technologies. I think that maybe the shorter term applications are things like diagnostics or sensors, right? Those things maybe just even from an approval standpoint are relatively simpler to get to real applications. But I'm pretty optimistic. I think what's really made me optimistic is the fact that these pharmaceutical companies are really getting interested in this technology. I've had graduate students that have and postdoctoral researchers that have gone, recently to work for Pfizer or other small biotechnology companies because there is real interest in this DNA nanotechnology.
So Carlos, I mean, my sense is for every kind of drug, maybe eventually it's going to be encapsulated in some kind of nano machine to improve delivery, to reduce resistance, to target better. If you have a neural drug, maybe you don't want to target it everywhere. it might be something that's will explode in a sense as a technology for every kind of anything you ingest, essentially. Anything you get, I mean, any therapeutic pharmaceutical that gets into your body.
Yeah, no, I tend to agree. You know, of course, I'm biased because I'm a nanotechnologist, but I do tend to agree. You know, there are several clinically approved nanotechnologies. Of course, you know, the COVID vaccine, an example of a lipid nanoparticle. But there are, you know, chemotherapeutics, right? There's things like, I think it's called Doxil. And then there's another one called what's it called? Vixios, I think, is another nanoparticle that has a cocktail of drugs in there. Right. So there are, you know, several examples of clinically approved sort of nanoparticle drug delivery systems. And I agree with you that I think it's only going to increase because of the advantages, like you mentioned. Oftentimes, the challenge of treating a drug is not killing the cell, right? You know, there are many, many drugs that can kill a cell, right? The challenge is killing the target cells without killing everything else, right? And that's really where nanotechnologies can be beneficial.